So we're all breathing, hopefully, and we don't think a whole lot about it. You know, we do it every day about 20 times a minute. Don't give it much thought. This guy's thinking about it a lot. He's up on the top of Mount Everest, and at that altitude, you actually can't survive very long without some kind of external oxygen supply. There's about a third as much oxygen there as there is here where we are, here near sea level in Denmark. So the amount of oxygen matters. And NASA, I don't know exactly how they did this study, but they did, where they tried to figure out just how much oxygen humans need and how long can they survive at the lowest amounts of oxygen that they require. And if you look here, we have, sorry, I need to get used to this pointer just a little bit. Unclimatized people and climatized people. And this is here, the amount of oxygen there is at sea level. And you can see that when oxygen levels dip about 50% or lower than what we have now, even people that are acclimated to low oxygen levels can't survive for very long. So the amount of oxygen matters. It matters to us. It matters to most animals. It hasn't always been this way. There hasn't always been this amount of oxygen that we have in the atmosphere now. And this is a story that I want to take you through. So hopefully that you can understand and appreciate a little bit about the history of oxygen on Earth and how that history has impacted the evolution of animals and ultimately, of course, the evolution of us. In order to look at a story like this, we need to understand something about geologic time. Many of you probably know this, some of you may not. The Earth is a little over 4.5 billion years old. That's about a third of the age of the universe. So is it young or old? I'm not sure, but I think it's pretty old. And when we talk about Earth time, we break Earth into various compartments. There's a time period here called the Hadean. About the first 600 or 700 million years of Earth history we know very little about because there aren't any rocks preserved from that time. Partly that's because there weren't very many produced, and partly it's because the ravages of time had destroyed the rocks that were there. Then we come into a period of time here called the Archean. We have an idea that life evolved at least by about 3.8 billion years ago. And this is a time where prokaryotes, bacteria, single-cell organisms ruled the world. Of course, there was certainly a great deal of evolution during this time, but it was a time of small organisms. We now move up into the Proterozoic, and during the Proterozoic, we're going to be spending quite a bit of time in this interval as we go along. This is the time when eukaryotic organisms evolved, when multicellular organisms evolved, when animals first evolved. Not all at the same time. There was a progression in this, but this is a very important time in Earth history. And then we come to the Phanerozoic, the last 540 million years or so. This is the time where animal ecosystems became dominant. This is a time when plants evolved. This is a time when the Earth evolved into a state that we recognize today. So, how do we know about oxygen? How do we know anything about the evolution of oxygen through time on Earth? Well, we look at rocks. That's how we understand it. And here we go back in time to about 2.6 or so billion years ago. We're looking at some rocks from Australia. And these are rocks that were deposited on the seabed, the bottom of the ocean. And they're very different from the kind of rocks that deposit on the bottom of the ocean today. At this period of time, it wasn't uncommon to find these types of rocks. What's particular about them? They're red. They're full of iron. These rocks are chock full of iron. And the only way that you can deposit iron-rich sediments around the world is if iron is free to distribute itself in the bottom of the ocean. Well, you probably are aware of this in one way or another, but iron and oxygen actually don't like each other very much. They form into rust rather pretty quickly if you try to mix a dissolved iron phase with oxygen. So in order to transport iron, and for those chemists in you in the 2+ state, through the bottom of the ocean had to be oxygen-free. So we're talking about vast expanses of the ocean at that period of time that didn't have any oxygen. And that was because the atmosphere itself had much less oxygen in it. So this is direct evidence that oxygen levels were much lower then than they were today. But there are other ways that we can look at this too. Does anybody recognize this mineral? This is pyrite, FeS2. It's an iron sulfide mineral. And you can do this at home. It's actually kind of a neat experiment. You can go to a rock shop somewhere, maybe at the Museum of Geology, the Geological Museum here, and buy a cube of pyrite. And what I'd like you to do if you do that, maybe buy two, one to keep to look at because it's pretty, and one to put outside in your garden, and just see what happens to it over time. I will bet that over some time, perhaps it takes a year, maybe it takes a couple of years, that cube of pyrite is going to rust away. It's going to rust into nothing after a period of time. And that's because this pyrite will react with the oxygen in the atmosphere. Now with that knowledge, we're going to go and we're going to look at some rocks again. And now we're looking at 2.7 billion-year-old rocks from South America. And these rocks are full of pyrite. And this is detrital pyrite. This means this is pyrite that has been moved. This is, by the way, this is a bottom of a river. We're looking at a river sediment. And these pyrite grains have been moved along with the flowing water of the river, and they have deposited together with these other cobbles and stones and pebbles in the river's bottom. Well, that doesn't really happen today. And the reason that it doesn't happen today is because the pyrite that gets exposed on land becomes oxidized with oxygen. So this is more evidence that the Earth at this period of time had very, very low oxygen levels. How low? Okay, now we come to the next piece of evidence, and this is probably the deal breaker. This is the one that really matters the most in our understanding of the history of oxygen on the Earth, at least this early part of the history. And in order to talk about this, I need to talk about isotopes. And I know, I mean, God, some people really, you mention the word isotopes, and a lot of people just, whoops, they turn right off. But try not to do that, because this actually is not a difficult story. It's not a complicated story. So, sulfur has 4 major isotopes. They're 32, 33, 34, and 36. Okay? Same number of protons, different numbers of neutrons for the chemists. Now, what's interesting about this is that there are various processes, some biological, some not, that fractionate the isotopes. What that means is these processes prefer some of the isotopes over the others, usually for thermodynamic reasons. One of those processes is a microbial process called sulfate reduction, where microorganisms take sulfate from seawater and they produce sulfide while consuming organic matter. A little bit like what we do with organic matter and oxygen, except they use sulfate. In that process, there's a fractionation. What happens is the organisms prefer the 32 a little bit more than the 34. Now, what's interesting about these isotopes, these other isotopes, is that typically when we have fractionations that are called mass dependent, the fractionations depend on the mass. So what that means is that if we have a fractionation between 32 and 34 sulfur, that the fractionation between 32 and 33 will be half of that, and the fractionation between 32 and 36 will be twice of that, and it depends on the difference of the mass. That's mass-dependent fractionation. Now, when we go, well, let's not go back in time. Let's just look at the last 2.3 or 4 billion years to about this point. This delta 33s is telling us basically whether or not we have mass-dependent or mass-independent fractionation. And when we're on the zero line here, that means everything is behaving like we expect, like it's supposed to. We have mass-dependent fractionation. But look what happens when we go back here into this time period we've been talking about before. Now we're back around 2.4 and earlier time. Then everything goes hay-wacky. We no longer have mass-dependent fractionations. The fractionations become mass-independent. That's weird. Why is that? Well, that is because the sulfur at this point in Earth history is mainly coming into the surface environment of the Earth through volcanoes as SO2 gas. This SO2 gas undergoes photochemical reactions in the atmosphere with ultraviolet light. And those photochemical reactions produce a mass-independent fractionation. Okay, now, the cool part of the story is that this wavelength of UV light is blocked by ozone. So if you have ozone present in the atmosphere, then that fractionation is at least well reduced. So what we have here is an ozone meter, basically. Before this time in Earth history, we didn't have ozone in the atmosphere. After this time, we did. Where does ozone come from? It comes from oxygen. So what we're looking at is an increase in the oxygen content in the atmosphere in something called the Great Oxidation Event, or the GOE. So that's pretty cool. We can get a direct record from the chemistry of the rocks of when there was a dramatic rise or change in the concentration of oxygen in the atmosphere based on these isotopes. If we look real closely at this record, and this is a record from South Africa, and we're looking at time, and we can see here we have mass-dependent or independent, mass-dependent fractionations, then become mass-dependent. Here are those detrital pyrites I was telling you about. You can see that when they're depositing, we get this wacky sulfur isotope behavior. And then we have something called continental red beds. They're very red, iron oxide-rich sediments that deposit or form on the continents, and they happen in an oxygenated atmosphere. So you can see that we can see expressed in the rocks the expression of this change in oxygen we see in the isotopes. How much oxygen does that mean? Well, I guess I'll show you that later, but I'll tell you just now. Calculations suggest that with a mass-dependent fractionation, we're talking about mass-independent fractionation, we're talking about oxygen levels about 100,000 times less than what we have in the atmosphere today. So what that change indicates is a change from oxygen levels which are super low to oxygen levels which are considerably higher than that. And we'll talk about how much higher in just a little while. So where does oxygen come from? It comes from these guys, cyanobacteria. Cyanobacteria were the first organisms on Earth to make oxygen. They are the heroes of the story, and they're actually one of the real heroes with respect to the evolution of life on Earth. Without these guys having evolved first, we would not be here. So cyanobacteria are our friends. There's no question about that. And if you were to go back in time to look at an Earth that contains cyanobacteria, but not the higher life forms that we're used to today, it might look something like this. Have you ever been to Australia? If you ever have, take a trip out to West Australia, go to Sharks Bay. It's a fantastic place. And if you do that, you will see these stromatolites, which are created by cyanobacteria, which are precipitating calcium carbonate as the structure. So if you close your eyes and think about it, the Earth 2.5 billion years ago might have looked a lot like that, which I think is kind of a cool image, kind of a cool way to think about it. So, cyanobacteria make oxygen. But that isn't necessarily why we have oxygen in the atmosphere. Of course, originally it was the source of oxygen, but the question really is, how do we accumulate oxygen in the atmosphere? How do we get it in the atmosphere? How do we get lots of oxygen in the atmosphere? So if you think about it like this, if you have a plant in a closed container, and that plant produces oxygen, in the normal course of things, almost all of that oxygen will be consumed again by respiration, either by respiration from the plant or by respiration from the microbes that are living in the soil where the plant is growing. So the balance is really close. What that means is that you don't actually necessarily accumulate a lot of oxygen in a system that is closed like that. Or another way to think about it is that in a system like I'm discussing, the amount of oxygen that you accumulate in the atmosphere of that is equivalent to the amount of organic matter, of plant organic matter, that doesn't get decomposed. That's how much oxygen you get. It's not how much oxygen is produced during the day, it's how much organic matter doesn't get oxidized again. And that's the way it works on Earth. So we have oxygen produced by, let's go back in time, cyanobacteria, now it's algae and plants. That produces oxygen. The amount that accumulates into the atmosphere depends on the amount of organic carbon and pyrite burial. I won't talk too much about that. Just think about organic matter. That's probably the easiest way to think about it. It gets buried in sediments. So the amount of organic matter that gets buried in sediments tells you how much oxygen actually gets distributed into the atmosphere. Not how much is produced, how much organic matter gets buried. Then what happens to the oxygen? Why doesn't it just keep accumulating forever and ever? Well, it doesn't, because that oxygen reacts with rocks, organic matter, the pyrite that we've talked about, the rocks that get thrown up back onto the continents through the rock cycle, and it gets used that way. And also, there's this funny thing about volcanoes, volcanic outgassing. Volcanoes are constantly distributing compounds like hydrogen gas and SO2 that get utilized and also form an oxygen sink. So that's how we have oxygen in the atmosphere. I hope that's clear to everybody. I hope that it is, because it's not just how much is produced, it's how much organic matter that actually gets buried in the sediments that determines how much oxygen we get produced or delivered into the atmosphere. About the history of oxygen, we've talked about this increase of oxygen here at about 2.3, 2.4 billion years ago. Our best guess is that oxygen increased by about 3 or 4 orders of magnitude to levels which are somewhat uncertain. It's a whole nother talk. I'm not going to go into that. There might have been some large changes in that. But this increase here turns out to be super important. Super important with respect to the evolution of life on Earth. And I'll try to explain why. Now we're talking about an oxygenated world. And we're going to start to talk about moving from this earlier world with cyanobacteria and other bacteria, prokaryotic organisms, into a world that's a little bit more closely related. A world that starts to reflect a little bit the world that we're more familiar with. So one question is, when and how did eukaryotic organisms evolve? In the broad spectrum of things, there are 2 types of organisms on Earth. There are prokaryotic organisms, which I've been talking about, and then there are eukaryotic organisms. Eukaryotic organisms we're eukaryotic organisms. They're cells that contain nucleus, and they contain organelles. They contain membrane-bound bodies that the prokaryotes don't have. They're more complicated than prokaryotes. So there are a lot of ideas about, well, not there are a lot of ideas, there's some good ideas as to where eukaryotic organisms actually came from. And there's sort of the present best understanding of it is that eukaryotic organisms, basically our ancestors, come from a symbiotic relationship between different types of earlier existing prokaryotic organisms. Specifically, there is a group of prokaryotes called archaea. And this group of archaea are a little bit more closely related to eukaryotes than other groups of prokaryotes. And the idea is that a group within what is known as the Asgard archaea, the membranes of this cell began to develop some kind of a fluidity. And that fluidity allowed them to engulf an organism of a group called the alpha proteobacteria. Now, what's important is this alpha proteobacteria is an aerobic organism. This bacterium couldn't have existed before oxygen accumulated into the atmosphere. So the evolution of eukaryotes is fundamentally coupled to that increase of oxygen in the atmosphere that happened about 2.3 billion years ago. I'm not saying it happened just then. It probably happened quite a bit later. But if that didn't happen, then eukaryotes wouldn't have evolved. So this alpha proteobacteria is engulfed by the Asgard bacteria into a symbiotic relationship. It's producing energy for the Asgard archaea. And then at some point, it becomes the mitochondria, the energy broker of the cell. At that point, the fundamental eukaryotic cell is established, and then the eukaryotic lineage breaks off into all kinds of other types of eukaryotes that we're familiar with, plants and animals eventually, and all sorts of things. But this is the series of events that sparked the initiation of eukaryote evolution on Earth. And again, it required oxygen in the atmosphere. How much oxygen did it require? Now, that's an interesting question. So there's not a whole lot of work actually done on trying to understand just how much oxygen these early eukaryotes might have needed. But we've tried to address that in a way by looking at how much oxygen would have early algae needed. And you say, what? Why do algae need oxygen? Algae produce oxygen. Why in the world should they have any kind of a need for oxygen? Well, they do. And the reason is because in the typical life cycle of an algae, they produce oxygen during the day, but then during the night is when they actually reproduce. They divide, and they make new DNA. They conduct a lot of their very important cellular processes. Now, why do they do that at night? Well, they do that at night to protect themselves, and we'll get into this much more later on, against the bad side of oxygen metabolism. And just to explain it a little bit. So we respire oxygen, and that's good. That's how we live. But the process also produces something called reactive oxygen species, which are damaging to our cells. And we'll talk a lot more about that again later on. And algae have figured out if they conduct these processes of DNA replication and cell replication at night, that they reduce the possibility of damage that way. So yes, most algae have a very important requirement for oxygen. So what we did is we wanted to see just how much do they need. So we took algae, we grew them up in cultures that varied in oxygen concentrations between 1% of what we have in the atmosphere today and 100%. And what we found was something very interesting. Green algae, which is supposed to be a very ancient group of algae, couldn't do anything, hardly, when there wasn't any oxygen there. They couldn't grow. They needed to have some oxygen available in the environment in order to grow. But as soon as they got 1%, that was fine. That was enough for them. then they were happy. Curiously, red algae, which is the other ancient group of algae, some of them actually did quite okay, even with zero oxygen. And one of the things we want to try to understand is how and why they did that. How in the world were they able to survive without any oxygen? We don't really know that yet. We're going to figure it out, but we don't really know that. Some of them were affected. So I guess if one was to try to summarize this, the amount of oxygen that the early eukaryotes needed was probably something like about 1% of what we have in the atmosphere today. That's not a lot, but it was enough, probably, for early life. So that's the levels that oxygen needed to approach in order for this early eukaryotic life to be able to have survived. Okay. So we're racing a little bit here, but I want to now bring you into a discussion of animals. That's how we started this talk. You know, we need a lot of oxygen. We need about half as much as there is in the atmosphere in order to live. How about the first animals? How much oxygen might have they needed? Well, in order to do that, in order to discuss that, let's talk a little bit about where animals came from. So, current models suggest that animals evolved from a group of organisms called choanoflagellates. They're our closest sister group, or non-animal sister group. What's a choanoflagellate? A choanoflagellate is a protist that lives in the environment. It eats by a process called phagocytosis. That means that it has this flagellum which beats, and it has this net behind it, and it beats, and that creates a current, and the current causes bacteria and other things, prokaryotes, to get caught in the net, and then it consumes those by engulfing the food around through its cell membrane. That's how it lives. Now, choanoflagellates have a particularly interesting thing, at least some of them do, where they occasionally form colonies like this. And it's not completely known exactly why they do this, but they do. One reason to form colonies would be as protection. So if you wanted to not get eaten by another bigger protist, it might be an idea to form a colony with your buddies so that you become a bigger object to eat. Another idea is that perhaps this colony is better at circulating food into the environment of the colony itself. So maybe it's more efficient at feeding with a larger colony like this. That's a possibility too. We don't really know what the specific advantage is, but this is where you have to squint a little bit. You know, I teach in my lectures. Let's say the origin of life. You really have to squint a lot if you want to talk about the origin of life. You have to really talk about the big ideas rather than trying to get down into the details, because nobody knows anything about the details, and it's kind of the same here. But single-celled organisms are single-celled organisms. They're all of the cells in a particular population do the same thing, right? Multicellular organisms like us have cells which have multiple functions. So even though we start with the same cells, we develop cells that have lots of different functions that they do. So the idea is, how do you go from a single-celled organism with one function to a multi-celled organism with multiple functions? And the idea is that you start with a colony. And in a colony, what can happen is that maybe one cell, through evolution, mutation, develops a slightly different type of a function for the organism compared to the others. And that particular type of function is advantageous to the organism so that it becomes part of the genetic properties of this colony and so forth. I mean, again, you have to squint if you want to really think about this, but if you squint and do it, you can sort of imagine that you can go from a single-celled organism to a colonial group of organisms to something which is a multicellular organism with cells having different types of functions. That's sort of how we imagine it happened, and we figured it happened about 700 or 800 million years ago. So one of the things that we've been fascinated about in our lab is to try to understand, well, how much oxygen do these guys need? How much oxygen might the earliest animals have needed? Well, we don't have any of the earliest animals around, obviously, because that was 700 or 800 million years ago, but we do have their ancestors. So what you can do is you can look at the DNA content of organisms and you can compare it. And organisms that are closely related have DNA which is very similar, and organisms that are more distantly related have DNA which is more different. So what you do is you take all of this information, you shove it into a computer, and what the computer will give you are sort of comparative distant relationships between different organisms, and it's known as a phylogeny. And that's what this is. It's just sort of a textbook phylogeny of the history of life, of animal life, based on the comparison of different molecular sequences. What you find out is that the deepest branching organisms typically in this phylogeny turn out to be sponges. Sea sponges, sponges that live in the sea. And not every phylogeny does, most of them do. And actually there's kind of an interesting, I haven't really have a slide that shows this, I should have done that. But the inside of a sponge doesn't look so different from this organism here. The pumping part of a sponge, the way the sponge actually generates water flow through its body, is through a cell type called a choanocyte, and the choanocyte looks every bit like a choanoflagellate. So you can sort of imagine that that relationship has carried on in sponges. But anyway, what I want to say is that sponges are deeply rooted animals, and you find them in the geologic record. This one's a little bit highly debated, but about 600 million years ago, there's a single fossil of an organism from China, which looks every bit like a sponge as we understand sponges. And then about 550 million years ago, there's another better preserved fossil, relatively newly published, which looks a lot like a sponge. So there's something that we find in the fossil record. So we believe that looking at sponges can tell us something about what the early animals might have been like in terms of their oxygen requirements and in terms of their general lifestyle. So this cute little fellow is called Tethya wilhelma. And I tell you a little bit about sponges. So I suppose you guys, many of you guys probably own dogs or cats or goldfish or something like that, right? And they're relatively easy to keep. You walk your dog a few times a day and you feed it every day, it's fine. The same with a cat. Same with a goldfish. You don't have to walk the goldfish, but obviously, if you feed it, it's okay. Sponges are really picky. They're really hard to keep happy. They're primitive, but difficult. And this particular sponge, Tethya wilhelma, was found living in an aquarium in the United States, and it was easy to grow, it's easy to maintain, and it's turned out to be kind of the lab rat of sponge research. So we decided we would look at it too. And the first thing that you can do with a sponge like this is you can figure out, well, how does it behave with oxygen? How does it respire oxygen? And what you do is you do kind of a simple experiment where you take the sponge and you put it into a closed container and you watch the rate with which the sponge uses oxygen. And then at a particular point, the sponge starts to become sensitive to the amount of oxygen that's there. That means it changes its rate of consuming the oxygen. And that happens here when we start to see a drop in the rate. So this is oxygen concentration. We start to see a drop in the rate. Now the sponge is starting to become sensitive to the amount of oxygen that's there. And if we do something called a Michaelis-Menten kinetic analysis of this, we find out that the oxygen level when the sponge is respiring at half of its maximum rate, that's called the Km value, is about 5% of atmospheric levels. That's pretty low. Okay, 5%, half its rate. But Don, what is that telling me about whether the sponge is happy or not? Is it living well? I mean, it's obviously being impacted somewhat. And I'd say that's a good question. So we decided to look at this in a little bit more detail. So one of the things which is really cool about this particular sponge is that it has a behavior. I want to show you this. I hope it works. Ah, damn it. That's really annoying. It's not working at all. Well, there it is. Okay. See what it's doing? About every 5 or 6 hours, this is speeded up photography, it does this contraction, and then it increases again, and it does it. And we're going to talk a little bit later in the next few slides about why it's doing this. But it does it, and it's a behavior. And that's one of the things that attracted us to Tethya, is it has this really interesting behavior. Most sponges don't have such behavior. So you can ask yourself, if this has a behavior, it's useful behavior, and if it's useful behavior and it stops doing it, then maybe that's not good. So what we did is we monitored the sponge with respect to this behavior related to the oxygen concentration. And what we found was that when the oxygen levels came down lower than about 5%, it stopped doing this. It stopped doing this contraction. So it was obviously being impacted by the oxygen below this. Was it unhappy? Maybe. It's hard to ask a sponge if it's unhappy. But there's one observation I'd like to show you which suggests that it might be able to tolerate a little bit lower than this. This is a photograph of a sponge which is held at about 0.5% atmospheric levels, and this one was held at full oxygenation. And we let the sponge sit in those situations for a couple of weeks. And even though the sponge wasn't doing that contraction thing, it's not dying either. I mean, how do you know if a sponge is dead? Well, it gets all kind of black and gooey, so you can sort of tell. And so, well, 4 or 5% seems to be when it changed its behavior. It might be able to live at even lower than that. But if you're thinking about a number, think 4 or 5%. That's probably where animals start to become impacted, at least early animals, by oxygen. It's a little bit more than what the algae need, but it's still quite a bit less than what we need. So early animals didn't need anything like the amount of oxygen that we do. And our best guess, not guess really, our best scientific results, even though there's a lot of discussion about it, suggest that that's the kind of oxygen level we were talking about when animals were first evolving. So that's good. Animals had enough oxygen in order to evolve. So I'm going to change gears just a little bit and explore a little bit in more detail the relationship between oxygen and animals. And I think this is stuff we're working on now, and I think it's super fascinating. In a sense, it's a little bit detached from the question I've just been answering, but hopefully you'll find it equally as interesting. So there's one thing to understand about how a sponge is relating to the external oxygen in the environment. We've just talked about that. How does its behavior change? How does its metabolism change? How is it impacted? But one thing we know about sponges is they contract. Remember that contraction? Why do they do that? Why do they contract? Well, we aim to try to understand why they do that. And in order to do it, we took a different sponge, our sort of local, one on a local team, Halichondria panicea, which you can find around here, certainly in the harbors in Copenhagen. And we grew it on a slide. So you take a little piece of the sponge and you attach it to a slide, but the slide has a light-sensitive fluorescent dye on it, so which is sensitive to oxygen. And what we can do is we can monitor then the oxygen concentration at the very base of this, we call it X-plant. And this is what we find. This is over cycles of about 10 hours or something like this. The thing goes anoxic inside. So oxygenated, anoxic, oxygenated, anoxic. The sponge is pumping water through its system, fully oxygenated, then it stops. That's a contraction. And when it stops, contracts, then the inside body is going anoxic. Certainly that's what Tethya was also doing when it was contracting. So it has this really cool behavior where it goes oxygenated and then stops. Oxygenated, deoxygenated. And as it turns out, this is really common for sponges. We look at another one, Haloclona rosea, another sponge on the local team. And Haloclona does it a little bit differently. Instead of having this sort of whole body contraction, this whole body anoxia that Halochondria has, it has a different style. What it's doing is it's sort of running these anoxic events throughout the sponge. So it's like this moving light show, which is really quite beautiful. Really very interesting. But it still has the same function in a way in that it's driving anoxia within the body. And it's doing it on some kind of a semi-periodic scale. So this is the area of the sponge which is anoxic, and then this is the extent of anoxia, the area which has had reduced oxygen. And it's not as frequent or as intense as Halochondria, but it's still doing it. And observations that other people have made with something called a microelectrode. You can actually make a glass electrode and you can poke it into a sponge and you can look at the internal oxygen level. It's very common for sponges actually to go anoxic. So why do they do this? So we don't really know, but we're going to give you some ideas as to what we think. Service the microbiome. So like us, sponges have a microbiome. They have a microbial internal system which is helping them with their livelihood. And the microbiome does all sorts of things. It cleanses out toxic chemicals, for example, or produces other things, vitamins that the sponge needs. So they have a microbiome. And one hypothesis is that this microbiome might need anoxia now and again in order to survive. So that's an idea, just like ours does actually in the gut. So it could be that that's one reason why they do that. Sponges don't have stem cells in the same way that we do, but they have cells which act a little bit like stem cells. And our stem cells propagate under low oxygen environments in the body. So it could be that that's when the sponge generates stem cells in order to regenerate its own body. And then we're back to this oxidative stress idea. It's possible that the sponge is doing this in order to repair damage from the oxidative stress of metabolism at high oxygen levels. The sponge actually, when it's pumping normally, is pumping fully oxygenated water through its body. Let's contrast with us. This is something that I've been working with an illustrator on to try to document and plot the oxygen levels within inside our body, the human body. This is full air, full oxygenated body. It's white. And you can see by the color scheme that most of our internal organisms are at much reduced levels of oxygen compared to what there is in the atmosphere. The brain is at about 30%. Let's see, the lung is actually at about 60%. Under the skin, it's almost anoxic. Of course, the internal gut, the external part of the gut is at about 40%, but the internal bleeds into anoxia when you get into the central part of the intestine. Interesting, the cell, the mitochondria, where energy is made, where oxygen is used in our body, there's almost no oxygen. It's about 1% of the amount that there is in the atmosphere. So where we are making energy actually in our cells, it's almost an anoxic environment. So even within our bodies, anoxia isn't so uncommon, and low oxygen conditions are actually very common. And why is that? Well, the prevailing idea is the same idea I was talking about with algae. Under reduced oxygen levels, there's reduced oxidative stress from these reactive oxygen species that are produced during our normal metabolism. You reduce the oxygen level, you reduce that problem. So we do it locally. I mean, we control it locally, and the sponge seems to be controlling it temporally. So that's kind of my favorite, anyway, working hypothesis is the sponges are conducting this anoxic turns off and on this switch in order to control and repair the oxidative damage from normal metabolism. Remember, they can't control the oxygen levels that's circulating through their body so easily unless they shut the circulation off. Like us who can do it all the time. So that's kind of a testable hypothesis. And we haven't really tested it yet, not really properly, but we've done some experiments which kind of hint at it. And one of those experiments is, well, what happens to the behavior of helochondria, this one, if we lower the oxygen levels? If this oxic-anoxic behavior is to repair oxidative stress, you might imagine that at some point when you lower oxygen low enough, it'll stop doing it. A little bit like tethia did, actually, but we don't have the internal oxygen dynamics of tethia to be able to express that. So we did that experiment with helochondria, not well enough, I must admit, but we did it. Oh. And anyway, this is 70% oxygen, where it's behaving as we just saw. When we get down to 35% oxygen, this is the same sponge. It's acting differently. It's not exhibiting the same kind of periodicity in the deoxygenation, and it's not as intense. And when we get to 7% or 15%, it's really so hard to see. I need to have a different scale for this. But the oxygen internally in the sponge is hardly moving at all. So this behavior would be consistent with the idea that sponges are conducting this switch off and on with their internal oxygen metabolism in order to repair their oxidative damage. And what's interesting is that at the time that sponges evolved, when oxygen levels were more like this, which you can't see here for some reason, they might not have done this at all. It could be that back when sponges evolved, that they didn't need to do an internal oxygen switch like they do now. So one consequence of that, which is something my former student, Emma Hanlon, has been proposing and pushing, is that actually the earliest animals were pretty well adapted to that low oxygen environment. That was an environment that they really could dig because the oxidative stress was actually not that significant. What was problematic was when oxygen increased. Then organisms like sponges and like mammals and other invertebrates had to develop internal ways of regulating the oxygen concentrations so that they could survive under these higher oxygen levels. Not that it's not beneficial. More oxygen also can produce more energy, but it was an adaptation to higher oxygen levels that animals had to go through, which I think is quite interesting. So we need a lot of oxygen, that's for sure. The amount of oxygen within our bodies is much, much lower than what there is in the atmosphere. And I think that hopefully you can see that through the history of the Earth, there has been a progressive change in the amount of oxygen in the atmosphere. And that progressive change has both impacted the evolution of life, and the evolution of life has actually impacted that change. And I think that's the message I would like to leave with you tonight. Thanks.